If you have thick, straight hair, the kind a comb slides through, the kind that fights a curl and wins, the kind that grows in strands measurably heavier than most hair on Earth, you are carrying one of the strongest signals of natural selection ever found in the human genome. Not one of the more interesting ones, not one of the more debated ones, one of the strongest. When geneticists scan all 3 billion letters of human DNA looking for places where something was actively favored, actively spread, actively pushed toward the top, this one piece of DNA keeps coming back at the very top of the list. It has been sitting near the top for almost 20 years, and almost nobody who carries it has any idea that it is there. It is a single letter, one position out of 3 billion. And depending on where your family comes from, the odds that you carry it are either overwhelming or close to zero. In Han Chinese populations, roughly 93% of people carry the changed version. Across northern East Asia, frequencies climb as high as 90%. In some Indigenous American populations, it is effectively universal. Everyone tested has it. Across Europe and across Africa, it is almost entirely absent. That is not a gentle gradient. That is one of the sharpest lines anywhere in the human genome. For 2 decades, the story of where that line came from looked settled. A place, a date, a reason. Then ancient DNA pulled a body out of the ground beside a lake in Sweden, 7,500 years old, in a person with no East Asian ancestry whatsoever, and that person was carrying it. We are going to follow this one letter from a mouse laboratory at Harvard Medical School to that lake in Sweden to a study of 6,000 people across 5 Latin American countries. And by the time we get to the end, the reason it spread is going to turn out to have almost nothing to do with hair. This channel digs into the history written inside the human body, real studies, named, dated, and checked against the papers themselves. If that is the kind of thing you want more of, subscribing genuinely helps. Here is the first thing that is going to reorganize how you think about this. There is no gene for thick hair. There is no gene for straight hair either. Nothing in your DNA sat down one day and decided the shape of a strand. What actually exists is a gene called EDAR, ectodisplacin A receptor. And EDAR is not a hair gene at all. It is a receptor, a protein antenna sitting on the surface of cells in a developing embryo, waiting for a signal. When that signal arrives, EDAR passes it inward, and a cascade of instructions fires. And the instructions do not build hair. They build something more general than hair. They build the small structures that grow out of the outer layer of the embryo, the ectoderm. Every hair follicle you have, every sweat gland, the ducts inside your breasts, the enamel-forming cells that shape the surfaces of your teeth. The developing embryo builds all of them with the same toolkit, from the same signal, running through the same receptor. Think of EDAR as a dimmer switch that was wired badly, or, depending on how you look at it, wired brilliantly. It is a single switch, but it is connected to more than one room. Turn it up, and the light changes in the hair, in the sweat glands, in the milk ducts, and in the teeth, all at once. You cannot adjust one of those rooms without touching the others, which means the moment you change EDAR, you are never changing just one thing. Hold on to that. It is the whole story. So what is the change? The technical name is RS3827760. In the DNA itself, it is a single letter swap at position 1109 of the gene, a T where there used to be a C. That one letter changes one amino acid in the finished protein. At position 370, a valine becomes an alanine. Geneticists write it as V370A. And it lands in a very specific part of the receptor. Not the outside where the signal arrives, the inside, a stretch called the death domain, which is the part that reaches into the cell and recruits the next protein in the chain. Change that one letter, and the receptor does not break. That is the counterintuitive part. It gets stronger. Laboratory work using reporter assays has shown that the altered receptor drives more downstream activation than the original. Measurably more signal, passed further into the cell. Geneticists call this a gain of function. The dimmer switch got turned up, and every room it is wired to felt it. So one letter in one embryo somewhere at some point in the deep past. And today, more than 1 billion living people carry it. Now, how do you find where something like that began? You cannot dig up the original person. There is no grave, no name, no bone. What you can do is read the DNA around the change in living people and let the arithmetic work backwards. Here is why that works. When a new mutation appears, it appears on one specific stretch of chromosome, one particular neighborhood of DNA with its own pattern of surrounding letters. Every descendant who inherits the mutation inherits that neighborhood along with it. Over generations, the neighborhood erodes, shuffled and trimmed a little more each time. So the size of the surviving block is a clock. A long, intact block means the change is young. A short, chewed-up one means it is old. In 2013, a team published the most complete answer anyone has produced. The paper is Kamberov and colleagues in the journal Cell out of Harvard Medical School, the Broad Institute, and Fudan University in Shanghai, with Pardis Sabeti, Clifford Tabin, Bruce Morgan, and Daniel Lieberman among the senior authors. They took 24 markers spanning roughly 139,000 letters of DNA around the change, mapped the haplotypes, the neighborhoods, and then ran the whole thing through more than 1 million simulations, asking a simple question. If this mutation started at a particular place and a particular time, and then spread the way populations actually spread, which starting conditions produce the pattern we see in living people today? The answer that came back from the strongest few thousand of those million simulations pointed to central China, approximately 30,000 years ago. Sit with that number for a second, because 30,000 years is not very long. Our species is somewhere around 300,000 years old. People had already reached Australia. The cave paintings at Chauvet were already on the walls. Nearly everything that makes a human was long finished by the time this letter changed. This is a very late addition to a very old story. So we have a place and a date, Central China, 30,000 years ago. And that is where the story sat, in textbook after textbook for years. Then the ancient DNA came in. In 2015, a team led out of David Reich's laboratory at Harvard published a genome-wide scan for natural selection in the journal Nature. Matheson and colleagues, 230 ancient West Eurasians who lived between roughly 6,500 and 300 years before the Common Era. It was, at the time, the largest ancient DNA dataset ever assembled. Among those samples was a group of hunter-gatherers from Motala in southern Sweden. Mesolithic people, roughly 7,500 years old. They fished a lake, they buried their dead, and they had no East Asian ancestry. None, by every measure the team could apply. Motala sits on a narrow stretch of water in Östergötland, where a river runs out of a lake. The people buried there left behind fishing gear, worked bone, and a set of skulls mounted on stakes that archaeologists are still arguing about. They lived by that water for centuries. Nothing about them, in the ground or in their genomes, points anywhere near East Asia. 3 of the 6 Motala individuals were carrying the derived EDAR allele. And not a look-alike, not a separate mutation that happened to land in the same spot. The researchers compared every genetic site within 150,000 letters of the change, and the DNA neighborhood around it in those Swedish hunter-gatherers was the same neighborhood you find in people in Beijing today. The same haplotype, the same inheritance. They found it twice more further along. 2 individuals from the Afanasyevo culture on the Eurasian steppe, dating to between 3,300 and 3,000 years before the Common Era. And 1 Scythian from 400 to 200 BCE. The authors did not soften what that implied. They wrote that the Motala people show no evidence of East Asian ancestry, and that this suggests the derived EDAR allele may not have originated in the main ancestral population of East Asians, as had previously been proposed. Mesolithic people, roughly 7,500 years old. They fished a lake, they buried their dead, and they had no East Asian ancestry. None, by every measure the team could apply. Motala sits on a narrow stretch of water in Östergötland, where a river runs out of a lake. The people buried there left behind fishing gear, worked bone, and a set of skulls mounted on stakes that archaeologists are still arguing about. They lived by that water for centuries. Nothing about them, in the ground or in their genomes, points anywhere near East Asia. 3 of the 6 Motala individuals were carrying the derived EDAR allele. And not a look-alike, not a separate mutation that happened to land in the same spot. The researchers compared every genetic site within 150,000 letters of the change, and the DNA neighborhood around it in those Swedish hunter-gatherers was the same neighborhood you find in people in Beijing today. The same haplotype, the same inheritance. They found it twice more further along. 2 individuals from the Afanasyevo culture on the Eurasian steppe, dating to between 3,300 and 3,000 years before the Common Era. And 1 Scythian from 400 to 200 BCE. The authors did not soften what that implied. They wrote that the Motala people show no evidence of East Asian ancestry, and that this suggests the derived EDAR allele may not have originated in the main ancestral population of East Asians, as had previously been proposed. Now, I want to be careful here, because this is exactly the point where a story like this gets oversold. This does not mean Kamberov's team was wrong. 2 very good papers are pointing in slightly different directions, and both of them are still standing. What ancient DNA established is that 30,000 years ago, the population map did not look anything like the population map today. The variant existed in people scattered far wider than its modern distribution suggests. Somewhere in the deep past, there were populations carrying this letter who left descendants in East Asia, and populations carrying this letter who left descendants beside a lake in Sweden, and we have only just started to be able to see them. The map you would draw of this trait today is not a map of where it came from. It is a map of who happened to have children. Which brings us to the part of this story that nobody expected, and the reason I wanted to make this episode. Go back to Kamberov's team in 2013. They had the date and the place. What they did not have was the reason. Something pushed this variant from 1 carrier to 1 billion in 30,000 years, and thick hair is a strange thing for natural selection to care about that much. So they did something unusual. Instead of only looking at humans, they built the mutation into a mouse. They engineered a knock-in mouse line carrying the human version of the change, the same letter, in the same position, in the mouse's own EDAR gene. And then they simply looked at what happened to the animal. The hair got thicker, exactly as predicted, exactly as it does in people. That much confirmed the association was real causation and not a coincidence of geography. But the mouse had 2 other changes that nobody had gone looking for. The mammary glands were different. The ductal branching inside them had changed, and the fat pad was smaller. And the sweat glands, the eccrine glands, the ones that produce watery sweat across the surface of the skin, there were more of them. Measurably, substantially more. That last one is where it stopped being a mouse story. The team went back to living people, to a Han Chinese cohort, and counted. Carriers of the variant had a higher number of active eccrine sweat glands than non-carriers. The mouse finding held in humans. So think about what that means for the hair. 30,000 years ago, in the warm, humid, subtropical interior of southern and central China, a population is living in a climate where the single hardest physiological problem you face is dumping heat, not conserving it, losing it. Humans are the sweating ape. It is genuinely one of our defining adaptations, the thing that lets a slow, weak primate run an antelope into the ground over a long enough afternoon. And in humid air, where sweat evaporates poorly, the number of glands you have to work with matters enormously. A mutation appears that turns up a developmental dimmer switch, and one of the rooms wired to that switch is sweat. If that is what happened, if cooling capacity is what natural selection was actually reaching for, then thick, straight hair was never the point at all. It was a passenger. It came along because it happened to be wired to the same switch. 1 billion people's hair was reshaped as a side effect of something happening in their skin. And I want to be honest about the confidence here, because the researchers themselves were. Kamberov's team wrote plainly in the paper that the direct biological significance and the potential adaptive role of this variant remain unclear. That sentence is in the abstract. They found what the variant does. They did not prove what it was selected for. The sweat hypothesis is the leading reading of the evidence, and it is a good one. The environment fits, the mechanism fits, and the human replication holds. But there are other candidates. Other researchers have argued for the mammary side, that changes in the milk ducts mattered under specific conditions. And the causal proof is a mouse. What we have in humans is association, plus one measurement that lined up. That is the honest version. It is still, to my mind, a far better story than the certain one. So let us follow the letter forward, because it did not stop moving. In 2013, Tan and colleagues published in the journal Human Genetics, asking whether this variant did something the earlier work had missed. Hair thickness was established. Tooth shape was established. Straightness had never been tested in East Asian populations, because every gene previously linked to straight hair had been found in Europeans. They tested it. The variant is associated with straight hair, which is why the trait we are talking about is not thick hair or straight hair, but both at once. They travel together because they come off the same switch. In 2015, Peng and colleagues, also in Human Genetics, went to the Uyghur population in northwestern China, a group with substantial ancestry from both sides of Eurasia, and found the variant reshaping the face as well. Further pleiotropy. More rooms on the same dimmer. And then there is the study I find hardest to argue with. Adhikari and colleagues in Nature Communications in 2016, working with the Candela cohort, 6,357 people recruited across Brazil, Colombia, Chile, Mexico, and Peru. On average, this cohort is roughly 48% European ancestry, 46% Native American, and 6% African, with enormous variation between individuals. The EDAR variant shows up in that cohort, doing exactly what it does everywhere else, shaping hair. It arrived in South America across Beringia tens of thousands of years ago in the ancestors of Indigenous American peoples. Then 5 centuries of migration, colonization, and intermarriage picked it up and moved it again into families whose grandparents came from Iberia and from West Africa. In one North American study of Latino cohorts, the variant was carried by around 47% of participants. The letter does not know whose family it is in. It never did. And here is where the dimmer switch shows its hand one last time. If you have watched the episode on shovel-shaped teeth, the scooped, ridged backs of the upper front incisors, you already know this gene. It is the same one, the same single letter. That scooping and this hair come off the same switch in the same embryo at the same moment, which is why they so often travel together in the same person. 2 completely different looking traits, one shared cause. If you have one, check for the other with your tongue. And EDAR is not even done. In 2020, a team publishing in the European Journal of Human Genetics described a second variant in the same gene, a different position, 380 rather than 370, and a different letter change entirely. They tested it in cultured cells. It turns the receptor up by roughly the same amount as the famous one. Same effect, same gene, arrived at independently. The 2 changes sit on completely different DNA neighborhoods, which means they are unrelated events. But this second variant tops out at around 5% in southern China, Vietnam, the Philippines, Malaysia, and Indonesia. It never swept. It shows none of the fierce selection signal that the first one does. 2 mutations, same gene, same functional result. One of them reached 1 billion people. The other stayed at 5% in the tropics and stopped. Whatever was being selected for, wherever the pressure was strongest, it was not in the South. And there is one more finding in that 2016 hair paper that I think quietly dismantles the whole premise of a trait belonging to anybody. Adhikari's team found a completely different gene affecting hair shape. PRSS53, a serine protease, an enzyme expressed in the hair follicle, in the inner root sheath. A different substitution in a different gene on a different chromosome, altering how that enzyme is processed and secreted. And it, too, shows evidence of recent natural selection in East Asia. 2 separate genes, 2 separate mutations, 2 separate selective histories, both of them making hair straighter. Straight hair is not one thing with one origin. It is an outcome that human biology has arrived at more than once by more than one road. So let us bring this all the way back to you. If you are watching this with thick, straight hair of your own, here is what you are actually carrying. And it is worth being precise because precision is the whole point. You are carrying one letter, a T where an ancestor of yours had a C. That letter is somewhere around 30,000 years old, younger than the peopling of Australia, younger than the oldest cave art in Europe, 250,000 years younger than our species. It is one of the most recent meaningful additions to the human body. It did not come from a nation. 30,000 years ago, there were no nations. There was no China, no Sweden, no Peru, no border, no flag, no census category. There were bands of people moving through landscapes and having children with the people they met. And it is not a badge of belonging. This is the part I want to be completely clear about, because the internet will tell you otherwise. Carrying this variant does not mean you are East Asian. It sits at near total frequency in Indigenous American populations who have been separated from East Asia for tens of thousands of years. It sits in roughly half of some Latin American cohorts. It sat in the mouths and on the heads of hunter-gatherers in Mesolithic Sweden 7,500 years ago, in people with no East Asian ancestry at all. And straight hair itself can be built by an entirely different gene in an entirely different way. It is a distribution, not an identity. A frequency map, not a bloodline. It tells you something real about the movement of populations, and nothing whatsoever about who you are. And the last thing, the thing I keep coming back to, it may never have been about your hair. The best reading of the best evidence we have is that 30,000 years ago, in a humid place, in a population trying to stay cool, something turned up a dimmer switch. And the switch was wired to more than one room. Your hair changed because your skin needed to breathe. The thing everyone can see turned out to be the side effect. The thing that mattered was invisible. And it still is. A few thousand extra glands doing quiet work on the surface of your skin every warm day of your life, and you have never once thought about them. A single letter. Older than any nation, older than any border, older than any word we currently use to sort people into groups. Not a mark of where you belong, a record of where people went, who they met, and what the weather was like when they got there. It is on 1 billion heads this morning, every one of them a distant cousin. Every one of them proof that it made it. And if you want more of the history written inside the human body, real papers, real dates, checked against the source, this is where we do that.